of this webinar. And we shall endeavor to have some candid conversations about how we can leverage on each other and do a lot of technical transfer and technical technology transfer. And on this call today, we are joined by the webinar. We are joined by Dr. Simon Agwale, who is the president and the CEO of Innovative Biotech in Nigeria and the US. He's also chair of the COVID-19 vaccine task team on the African Vaccine Manufacturing Initiative, and also a member of the West African Health Organization, ECOWAS Expert Committee on Access and Production of Vaccines in West Africa. Simon has a vision for the development and investigation of HIV vaccine constructs from prevalent Nigerian strains, which are relevant to Nigerians. He was a postdoc fellow at the Institute of Human Virology in Baltimore, US, and visiting scientists at various institutions in Brazil, Germany, UK, including the US Vaccine Research Center and the US CDC. Agwale was West Africa's representative on HIV for developing countries coordinating committee at European and Developing Countries Clinical Trials Program. He is a recipient of several scholarships, fellowships, grants from reputable organizations around the world, and he has authored and co-authored quite a number of notable publications. It gives me great pleasure to introduce Simon to speak to us. I first met Simon, who is a very good friend of mine as well. I first met him in Cape Town, South Africa, and we've continued to work together at AVMI. My name is Charles Kamau. I'm the co-chair of the Communications Working Group at the African Vaccine Manufacturing Initiative, and I am helped by the very able and capable Tolu Disu, who is the other co-chair in the Communications Working Group. Having said that, please, if someone has a question, please, in between as Simon continues to do the presentation, please just type it out in the chat box. And at the end of it all, once Simon has finished the presentation, I'll give people an opportunity to still ask more and more questions. Thank you very much. Please remain on mute and allow me to welcome Simon.
Well, thank you very much, Charles, for the introduction. Thank you, Melissa, for putting this very important meeting together, and AVMI team for coming up with this excellent initiative. Today, I'm going to be talking to us about our plans of bringing international vaccine manufacturing expertise to Nigeria. Melissa, this is not moving. Let's see.
Okay, I can see your mouse moving, Simon. Are you able to change the screen?
Okay, great. So it's working now. Okay, so as we all know, the development and manufacture of COVID and other vaccines in Africa creates a strategic long-term benefit for the continent to be pandemic ready. And it is well established that the ability to manufacture hundreds of millions to billions of doses requires the vaccine manufacturing capacity of the entire world. And according to Corey et al. In 2020, in their publication in Science, I quote, "There is an immediate need to fund the necessary biomanufacturing infrastructure, including the fill-finish steps that provide vial vaccine products for distribution." And as we all know, presently less than 1% of all the vaccines that are used in Africa are sourced from within Africa, a situation that makes the continent vulnerable to epidemics and pandemic. The recent African Union, African CDC virtual conference on expanding vaccine manufacturing set a goal that by 2040, 60% of the vaccines routinely administered on the continent should be manufactured in Africa itself. Innovative Biotech, therefore, is responding to this by establishing domestic vaccine manufacturing capability in Nigeria to serve the country and then potentially African community. Now, this is the current status of vaccine manufacturing in Africa. As you can see, there are 2-3 manufacturers in Africa that have end-to-end, that is producing the antigen and then doing the fill and finish. The first is the Pasteur Institute in Dakar that has been in business for decades now. They started production of yellow fever vaccines in 1938 using the traditional egg method. And their current capacity is about 7 million doses per annum. Then you have the Vaxera in Egypt that is producing the DTP family of vaccines. Again, the capacity is just about 100,000 doses per annum, which is basically for domestic use. Then you have the Pasteur Institute in Tunis that produces BCG vaccines. And then you have Pasteur Institute in Morocco that also produces the typhoid, influenza, rabies vaccines. And for fill finish, again, you have Biovac, which is a company that, South African company that is based in Cape Town. Basically, they have, all kinds of partnerships with various big companies for fill finish of their products. And some of these include hepatitis B and DTP family of vaccine, pneumonia, and so on. And more recently, we have the Aspen Pharmacare, which is a South African company that has a relationship with the Johnson & Johnson vaccines to do fill finish of their products in South Africa for Africa. And then you have the Ethiopian Public Health Institute that is planning to produce vaccines. Then in Nigeria, you have 2 companies, BioVaccines, that is planning to produce EPI vaccines, and then of course Innovative Biotech, which I'm going to present our plan to domesticate vaccine manufacturing in Nigeria. Now, our mission as a company is to build a new business area in Nigeria through the development of world-class vaccines, diagnostic products, and the provision of excellent clinical services. And our key objectives are to provide high quality and affordable vaccines across all critical disease segments, and to develop one of the first modern African-owned vaccine of international standards with world-class business leadership, and to deliver and maintain growth at a minimum of 10% over the next 10 years. So our strategy for development and manufacturing of vaccines in Nigeria is based on a stepwise approach. First, we plan together with our partners, build up a vaccine fill finish facility in Nigeria to quickly respond to epidemics, and then the national program of immunization vaccine needs. As a second step, and we're doing this simultaneously, the development of a multivalent COVID vaccine, which I'm going to present towards the end of this presentation, our strategy to rapidly produce a COVID vaccine, which Africa desperately needs now. And then we're using the virus-like particle as our platform technology because of its flexibility and the fact that 2 vaccines that use this platform technology have been in use for decades. So the safety profile of this platform is well established. So what then virus-like particles or VLPs? VLPs are structural mimics of the native virus, but they are non-infectious particles and they are unable to replicate or cause infection because they do not contain the genetic material of the virus, so they cannot replicate. The VLPs are ideal candidates for vaccine production because of their similarity to native viruses in structure, biochemical, and immunological composition. The VLPs are highly immunogenic due to their particulate nature and display of a dense array of surface antigens. And in the case of COVID, the spike protein. Now, VLP-based vaccines are exceedingly safe and suitable for all demographics. For example, children, adults, and elderly, and they have previously been used to produce 2 very successful vaccines, hepatitis B vaccines and then HPV vaccine. And you will know that hepatitis B vaccine has been in use for almost 4 to 5 decades now, and the HPV vaccine have been used for almost 2 decades now. So the safety records of this vaccine platform is well established. Now, manufacturing of COVID and other VLP-based vaccines can be rapidly attained using scalable suspension culture of insect or mammalian cells. Now, I will present briefly what or the vaccines that we have used the VLP to produce and our strategy of moving this forward to clinical trials and eventually the licensure. So I will start with the Ebola VLP candidate vaccine. Again, as you can see, we use the baculovirus system where we express the glycoprotein and the VP40 of the Ebola Zaire strain. And then the VLP were purified from the insect cell, which is the SF9 expression. And then you have your Ebola VLPs displaying both the glycoprotein and the VP40. So this particular vaccine expression was established and then verified by electron microscope, as you can see, the expression of the Ebola VLPs, and then confirmed by Western blot using antigen-specific sera. Now, the product was tested in animal model, in this case, mice, and then the data was presented during the Targeting Ebola International Conference in Paris, France, May 2015. So here you can see that blood samples were taken before the primary immunization on day 0, and then the second dose or booster dose of the vaccine was given on day 28. So samples were taken before the booster dose. As you can see, with 1 dose, again, this is a 10 mice per group experiment. The first group received 25 microgram of the vaccine without adjuvant, and then the second group received 25 microgram of the vaccine with our proprietary adjuvant. Then the third group received 50 microgram, and then the fourth group received 50 microgram with our adjuvant. So you can see significant improvement in immunogenicity of the vaccine using our proprietary adjuvant. And then after the boost, blood samples were taken on day 56. And then you can see again that, there was a boosting effect, with or without our adjuvant. So we use the same strategy to develop a HIV VLP candidate vaccine. And the first thing we did was to, look at the different subtypes. So carried out molecular epidemiology of HIV subtypes in the country to have an idea of the predominant subtype so that we can target that for vaccine development. And that particular study was published in Vaccine in 2002. So you can read the whole, the full article. So after that, we decided to randomly pick plasma serum samples from 12 individuals to look at how broad the neutralizing antibodies were. This again was published in 2011 in PLOS ONE. And then as you can see, 3 individuals had antibodies that were broadly neutralizing. And then what we did was to sequence these 3 viruses, the entire GP160, and then we codon optimize that, redesign to make it, rich in expression in mammalian cell. And then we deleted certain segments to expose the neutralizing epitopes. And as you can see, we express the glycoprotein 140 and GAC. And then this particular VLP was purified from insect cell. And then you can see the expression confirmed again by Western blots, and then, by electron microscopy, and then also confirmed by Western blot. So you can see the expression of the envelope and then the GAC proteins. So the serum from the humanized mice were tested for neutralization against different HIV subtypes. So you can see here that in addition to neutralizing, homologous neutralization, that is the vaccine was made from a what we call the circulating recombinant form O2AG, which is a recombinant, HIV recombinant between A and G, which is homologous. So there was clear neutralization. And then again, the blood sample was, plasma sample was tested against heterologous viruses like the Ugandan subtype D virus, and then the Thailand subtype AE, and then the Indian subtype C viruses. So there was neutralization of all these that. So we have shown here that the sera from immune mice had the ability to neutralize, not only homologous viruses, but also heterologous viruses. So finally, I'm going to talk about our strategy using the same VLP approach, but the 2 previous vaccines, the HIV Ebola that we developed, we use the insect cell, but in this case, we switch because it's always very difficult to purify, these VLPs from insect cells, and then we switch to mammalian cell, which is easier to purify. So as you can see, there are 4 major vaccine platforms that have been explored and used for COVID vaccine development. Now you have the, of course, the inactivated, where you grow the viruses, you inactivate, you vaccinate, and then they're broken down to specific epitopes. And all this is happening within the cytoplasm. Then you have the second vaccine platform, which is the protein-based vaccine. As you can see, broadly speaking, VLP vaccine is also considered as a protein vaccine. So here you vaccinate with your protein, and then it's broken down to epitopes, and antibodies are made against these epitopes, and then protect against the infection. The same thing happens here with the VLP, and then you have your epitopes that the broken down to epitopes, and then you do induce. So this before COVID, all the vaccines that are available utilize these two vaccine platforms. Now, it doesn't mean that these other platforms have not been in use. Both the viral vector platform and nucleic acid vector, nucleic acid vaccine platform have been in use for decades, but this is the first time that they are being licensed for use in humans. So you can see if you use the viral vector, of course, you can use, in this case, most people use the adenovirus inattenuated to deliver the genetic material to the cells and then to the nucleus. You can see the two previous platforms only utilize the cytoplasm. And then the viral vector with either non-replicating or replicating viral vector deliver their genetic material, which is transcribed to the RNA. And then if you use the naked DNA, it goes straight to the nucleus, which is also transcribed to RNA. And then if you use the messenger RNA vaccine, again, you have your messenger RNA in the cytoplasm, so it doesn't get to the nucleus. And then it is translated to the protein and then broken down to the epitope, and then you make the right immune response. Now, based on that, what have we done? I mean, if you want to make a new product, there should be a justification for making something that is new. So what we have done is not only, because if you look at all the vaccines, COVID vaccines that are in use today, except for the inactivated key virus, they only use the spike protein, and then the spike protein is also from the original Wuhan strain of the virus. So what we have done is that, because as we're going to see later on, the advantage of the VLP is that it allows you to simultaneously express other structural proteins, not only the spike protein. So what we have done is to not only use the original strain, but to use the South African strain, because the South African strain is the most difficult strain or variant to neutralize. So the advantage of what we have is that you have the ability to express multi-gene. So we are expressing all the structural genes, including the spike protein from South African strain. That is the envelope, the matrix, and then the nucleoprotein. Now, the expression again is verified using an electron microscope, and then you can see the spikes on the surface of VLPs, and then confirmed by Western blot using anti-spike specific antibody, as you can see, full-length spike protein. The spike protein, we introduced some modifications in the cleavage site to stabilize the spike protein so that the neutralizing epitopes will be exposed. So this shows us basically the production process that we have developed, and then which is you get your cells, and then you grow the cells using fermenters, and then you do your clarification, and then purification, and then you have your bulk antigen here. So this process, because for you to build a vaccine factory, you have to develop, that is the forward integration, you have to develop the process, so this process has been developed, and then our partner Merck is going to now design the VLP facility based on the process that has been developed. And then these bulk products can now be sent to any fill-finish facility anywhere in the world. We're in discussion with several organizations, including Saudi, Egypt, that already have a fill-finish facility. So we don't waste time. We don't have to wait to have a facility up and running in Nigeria before we produce this. So immunogenicity and neutralization of these products are ongoing. Right now, as we speak, 8 groups of, 8 mice per group have received this COVID VLP-based vaccine. And the data will be out by June, which is next month. Then the immunogenicity or serum antibodies will be determined by ELISA. And then we will take the immune sera. We have already made pseudoviruses against the major variants, which is the South African variant, the B1351, the Brazilian variant, the B1, and the UK variant, the B117, and then the original strain, which was first isolated in Washington. That has been included. We're working now, if time permits, to introduce and include the Indian strain into this panel. So by next month, we will test the immune sera from the South African variant that we use in our own vaccine, which is the 2nd generation vaccines, to see if it will neutralize all these variants, including the Indian variants. If not, if there's any of this variant that is not neutralized by that, the platform that we have is very flexible, so we can easily include whatever variant that is not neutralized and make a multivalent vaccine. So it's difficult now to get slots, whether you want to build a fill-finish facility, you want to create your cell line or do anything, because a lot of companies, there's a long queue. Everybody is expanding their facility, both for upstream, downstream processes, and so on. But we're fortunate because of our relationship with Merck. Again, BioReliance is a subsidiary of Merck here in the US. And then the next step is to create the master cell bank, and then the working cell bank. So what that does is that we will develop that cell line, cell bank, and then characterize, that will include the virus clearance, the lot release, the analytical chemistry, the binding, the potency, the physical testing, chemical testing, microbiology, and then the genetic toxicity. So this whole process is going to take about 3 months because we were lucky to get a slot by the end of next month. So by September, October, we should have our master cell bank that is ready to now transfer to a contract manufacturer to produce the GMP material for clinical trials and eventual rollout of the vaccine. So summary of the COVID-19, we have successfully expressed the spike protein of the original strain and the South African strain, which we believe is the right appropriate variant for a 2nd generation vaccine. And then this is because based on the data that has been published recently, it shows specifically that, sorry, I have to switch this noise. 1 minute.
To come back, let me just continue asking you that if you have questions, just write them in the chat.
I'm sorry, it's early here, so I'm still at home. So you have the kids. I'm sorry about that. So based on the data that has been, published in just last week in New England Journal, the AstraZeneca vaccine efficacy is, against the South African strain is just about 10%. And then Novavax is about 49%, which is below WHO threshold for implementation. And then Johnson & Johnson efficacy against the South African strain was about 57%. So you can see clearly the justification for making a second generation vaccine. Now, our goal here is to be in a position to conduct, once the data is out next month, we'll begin to develop the clinical trial protocol for phase 1, which we plan to do in parallel here in the US and in Nigeria, because one of the strategies that we have is to build local capacity to do all the phases of clinical trials in Nigeria, because that's where we are located. And then we have contracted a US manufacturer, a CMO, to produce the initial doses. We don't have to wait, because if we want to wait to have a factory, which is the next thing I'm going to present, it's going to take about, if you have not started, it's going to take you about at least 4 years to set up a functional, whether you're doing forward or backward integration, 4 years to have the facility up and running. So we can't wait for that. There's urgency in getting this product. So we have to work with a contract manufacturer to make this happen. So once this is ongoing, I mean, we're doing this in parallel, building the factory and then producing the vaccines using a contract manufacturer. And then we'll continue to be doing that until the facility is up and running, and then we can transfer the entire process for manufacturing in Nigeria. So now, building vaccine manufacturing facility, there are 2 ways to do this. The first is the bulk antigen production, which we have decided to do. And there are also 2 ways to do that, because one is when you have your technology, you start from the concept, the preclinical testing in animals, and then you do your clinical testing, phase 1, phase 2, phase 3, you license your product, and then you build a vaccine factory based on the process that you have developed. That's one aspect. The second aspect to produce antigen is to license the technology from another company. So you don't have to repeat all these steps that I've mentioned, but you go straight, just basically like what I presented. If we license the technology to you, for example, all you would do, you don't need to repeat all those things that we have, that have presented, because all that has been done. So we will send the cell line to you, and then send the SOPs, the protocol to you on the various. So you now build that facility based on the protocol that we have sent, to you. So you don't repeat all the preclinical testing, the concept preclinical, clinical trials, and so on. But some countries will require that you do what we call the bridging studies, which is, you have the original product and then what you have produced, and then look at the correlates of protection. Now, the second step the second way to build a vaccine factory is the formulation fill finish. So you leave this, you just set up a facility to do formulation fill finish. Both of them have advantages and disadvantages. The advantage of number one, where you produce your own antigen, is that, of course, it's more difficult from technical perspective, but more value capture. But in the second approach, it's technically less challenging, but capture less value, because you have to continue to rely on the partner to provide the bulk material. I mean, the typical example of bulk antigen production is AstraZeneca Serum Institute, Novavax Serum Institute. And then a typical example of formulation fill finish is Johnson & Johnson, and then the Aspen in South Africa. Now, the advantages of local manufacturing is the fact that you know, it will enable the sub-region to address the pandemic and future pandemic or epidemic. And the facility will also be used to produce vaccines for the sub-regional national program immunization. And then it will create an attractive platform for global vaccine companies to localize production and drive distribution market and share growth across the country. So you can see, because Serum Institute had the facility to produce this vaccine, it was natural for the companies, you know, to license the technology to them so they can produce for their, you know, people, and then for the developing world. So it will help train scientists in the sub-region in vaccine development and other vaccine-related activities, and then it will strengthen the supply and distribution channels and cold chain systems. So in summary, we have developed Ebola candidate vaccine that is highly immunogenic in mice. And then we have developed and evaluated an HIV VLP vaccine that induces broadly neutralizing antibodies in mice. And the primary immune response was boosted, unlike, you know, the viral, you know, vectors, because if you use the viral vector to deliver your antigen, you are not only inducing antibodies or immune response to the antigen, but to the backbone as well, which will impede the immunogenicity of the product down the line. Now, we've shown that our proprietary adjuvant enhance immunogenicity at both time points. Now, for both Ebola, HIV candidate vaccines, we didn't use any existing adjuvant, but in the COVID, as we are speaking now, the mice received the alum. Alum is an adjuvant that has been used previously in VLP vaccines. So we're comparing that head-to-head with our proprietary adjuvant to see which one will give us the best immune response, and that will be the one that we adopt moving to clinical trials. So we have developed COVID-19 VLP candidate vaccines, and immunogenicity neutralization assays are currently ongoing. And given the immediate need for COVID vaccines and the efficiency, safety, adaptability of our VLP platform technology, we are confident in producing a high quality product using the most cost-effective methodologies available. And then recombinant VLPs have inherent advantages in safety. Like I said, that the two vaccines, successful vaccines, hepatitis B and the HPV vaccines, have been used for decades. So this is basically critical for mass vaccination in Africa, because in Africa you have HIV, AIDS, and so on, and other immune disorders. So you need a vaccine platform that is safe in this population. And then finally, to establish domestic vaccine manufacturing capabilities in Africa to serve the country and then the continent. Of course, building, it's important to mention this. Everybody wants to build vaccine factory, you know, in Africa. Everybody wants to build vaccine. This is not, it's not rocket science, but it's also not a trivial thing. If not, you'll have seen vaccine factory all over the place. So you can't do this. We wouldn't have come to this level without the participation, collaboration, partnership with some of the best in the industry. We'd like to acknowledge Merck in Germany, without their platform, wouldn't have been where we are today. And as you can see, Millipore, Sigma, BioReliance, they are all subsidiaries of Merck. They have some of their staff that are dedicated to this project that they paid for, not from innovative biotech, because we have a good technology and they see that this is going to help the continent. And then we have technology partners like Technovax, Medigene, and our contract manufacturer, Vigene, here in the US. The good thing is that they have fermenters from 50 liters up to 500 liters. So before we have a factory up and running, apart from producing the initial clinical trial grades, once we do phase 1, phase 2 trials, of course, phase 1, like I said, is going to be done here in the US and in Nigeria in parallel. Phase 2, because it's South African strain that we use, a variant that we use to make this vaccine, we want to do phase 2 trials, phase 2AB, to have limited efficacy data from South Africa. And then phase 3 trials we're looking to do in South Africa, in Nigeria, and then in Brazil. So all these things would depend on the data that we get next month. Once the data is out, then we can now begin to develop the clinical trial protocol, which is the IND for submission to both the US FDA, Nigerian NAFDAC for phase 1 trial, and then we'll work on phase 2 protocol for South African, and then phase 3 protocol for South African, Brazil, and then Nigerian. And then I would here like to thank WAHO ECOWAS that have been strongly behind this initiative. They see this as a very important project for the sub-region. And again, of course, I have to thank the Nigerian government. And finally, you can't do this without funding. So we have one of the best financial advisors in this industry, Lions Health Investment in the UK, that is putting the financial structures together. So I thank you very much, and I'll be very happy to take questions.
Thank you very much for that lovely and quite important discussion. That and it's good to see that this is now happening, within Africa, and even as we look forward to increase our vaccine manufacturing capacity within the continent. There are quite a number of questions that have come in, but maybe just to start us off, one of the questions that I really wanted to ask is your general experience and what that has been, especially when you're talking to governments as a purchasers of your vaccines, how that process has generally been, and what are some of the other lessons that even as other manufacturers or other innovators want to engage with their governments, what has that relationship been?
Well, I think the first thing is basically to convince the government that you have a product and you have a product that works. For example, right now, because of our relationship with the big players, they are able to send information to other countries. So we have Saudi, we have Egypt that are interested in this technology platform. But I can assure you that by the time the data is out, because all this is without the data, when the data is out by next month, that is when, and this is what we are encouraging African leaders to do, that the only way you can get vaccine is to take that initial risk to place the order in advance before you know whether the product will work or not. Because if the data is out and shows that it's working, you have to join the queue. you know. So this is one lesson that I want African leaders to to understand.
Interesting. Thank you very much for that. Jean is asking which route of administration has been tested.
Intramuscular.
And Diana was asking a bit earlier, you know, what's the estimated timeline for the manufacturing facility to be up and running?
Well, like I said, we didn't want to focus on local vaccine manufacturing right now for COVID because it's an emergency. And that was why we had to look for contract manufacturers so that you can start producing the vaccine and then people start benefiting from it while you spend time to build the vaccine factory. And this is a very important question because for you to be part of to join the queue, now the queue, if you want to, let's say, for example, order equipment for fill finish, you have to wait for 8 months to have a slot. And then you can now start counting 18 months from that. So that's why I said that if you have not started, it will take you nothing less than 4 years to have your facility up and running. So we didn't want to wait for that, you know, because there are several contract manufacturers all over the place. So if you have your technology, then you can tech transfer. So the Vigin, our contract manufacturer, is the first company we're transferring the technology to. We can transfer to Saudi. They have identified interest. Egypt, they have also identified. So we can transfer this technology to various you know, companies around the world. But you know, while we're doing that, we're also building our facility in Nigeria.
Yeah, I mean, it's interesting, and you know, it's always, and I think that that's why it should be a pipeline approach, so that you think more down the road and start with, you know, what works. So I think you're right by pointing that out. Jean also had a few concerns about whether there have been provisions for either unusual thrombotic events and or antibody-dependent enhancement by native virus be included in the clinical trials.
Well, I mean, these are good questions. These are the things that we're going to look at. But you see, like I presented, this particular platform technology was used to develop a hepatitis B vaccine, which has been in use for almost 4 to 5 decades. So it has 40 years, 50 years history of safety. And then you have the HPV vaccine that has about 20, 30 years history of safety. But that doesn't mean that using the same platform to make a COVID vaccine automatically will translate to that safety record. But that is the record that we have. So I understand the point this you know, person is asking. Because there's this recent thing about antibody enhancement, that and all of that. So that will be included in the clinical trial protocol.
Yeah. So what plans are there to utilize a facility for production of other vaccines outside COVID-19 at the moment?
This is the point. I mean, the whole idea is not about, like, you know, like we stated, a full cycle manufacturing capacity is where you do the production of the antigens and then you do your fill and finish. So this is not only for COVID. I mean, like I presented, you use the same platform. You can make your hepatitis B vaccine. You can make your HPV vaccine, you know, using the same platform. You can make experimental vaccines like HIV, Ebola that are presented. And then for the fill finish part of it, you can fill any vaccine. You know, you can use that for your EPI vaccines because you are getting the bulk product from other manufacturers and then you're doing your fill and finish.
Yeah, so one of the other things is what will be the targeted manufacturing capacity, say doses per year of the Nigerian facility, and still the rough estimate of the sale price per dose?
Yeah, the price per dose is difficult to calculate now because right now, you know, we are doing what we call the process scale-up. So you have to know how many doses that you will make if you use 500 liter fermenters, you know, and then all the other costs, you know, associated with that. Then the capacity, the initial capacity that we are working on for Nigerian is about 400 million doses per annum. It's a modular facility, you know, so it can be easily expanded. We decided to start with this. You don't want to go and start making 1 billion doses of vaccines. You know, the off-takers, you know, so now COVID is, everybody needs COVID. So after COVID, what happens? You know, will that momentum remain and so on? So you have to be careful business-wide to take such decisions. So the initial capacity will be 400 doses of vaccines per annum.
I mean, so what size of, in terms of working volume and number of bioreactors will be used at the Nigerian facility?
This is the point. A good question. The right now, the production, even in its crude form, that we are getting right now is about 7 milligram protein per liter. So we're working to improve the media so that you can improve that production. So these steps have to be completed before you know the size of the fermenter that you will use. So the initial production using the contract manufacturer that we have right now that has up to, of course, initially for clinical trial, you know, material, we need only 100 liter fermenter, which they have. So the contract manufacturer has up to 500 liters. So we will learn a lot working with this contract manufacturer, and then we can now take our decision based on the size of the fermenter that we will have. But again, if you are building a facility of that magnitude, you are not building it like a fixed you know, fermenter to say, I'm putting only 500 liter fermenter. So you have to have different sizes of the fermenter. If somebody comes to you and says, I have, you know, a candidate vaccine against Lassa, for example, I need 3 million, I mean, I need 3,000 doses of the vaccine. You can go and use your 500 liters. So the person is looking at maybe 50 liter fermenter. So you build a facility that is flexible, that can accommodate any quantity of the vaccine that you need.
Yeah, I mean, you referenced making multivalent vaccines. Do you see this as a cost-effective strategy and target diseases of zoonotic origin?
Yes.
To be suitable for selection?
Good question. If you look at our strategy, I mean, the vehicle is very exciting platform because based on studies that have been done, if you use South African strain and you are able to neutralize South African strain, then that vaccine will be able to protect against all the other variants. But the plan B is that's why we made pseudoviruses from the various variants so that if there's any variant that is not neutralized by this South African strain, the platform we have, it's easy to incorporate that into that platform and produce another vaccine. So the same thing goes for any other vaccine of interest, you know, to us. So it's a very flexible platform that we can introduce, you know, antigen of our choice, and then depending on the customer's need and depending on the need of the continent as well.
You know, from all indications, 3 major vaccines are in the pipeline. And someone just wanted to know, you know, priorities in rolling out and what is the timeline, product by product?
Yes, the initial focus right now is COVID. You know, like I said from the presentation for this, we'll have by September, October, we'll have the cell line, the master cell line bank for COVID. And then that will be transferred to the contract manufacturer. So we're looking at third quarter of next year, of this year, sorry, to have clinical materials that we can do clinical trials. But for Ebola, HIV, immediately that goes forward. It's the same platform, the same time frame. So we can take, let's say, Ebola forward, and then create a cell line for Ebola, and so on. But right now, our focus, because of the need for COVID, is to focus on all the resources, energy that we have to focus on making a COVID vaccine.
In terms of technology to create these vaccines, how is mRNA-based technology different from VLP-based in terms of the efficacy and also, yeah?
Good question. I mean, like I presented, you can see the two vaccines platforms that are in use today, the viral vector vaccines and mRNA vaccines. They have been in use also for years, but this is the first time that they are being licensed as vaccines. So we don't know the long-term effects. We don't anticipate that there's going to be any problem in the future, but we can make a mRNA vaccine easily. But the key thing is that we are focused on what has been in use for years, well-established safety records, well-established efficacy data, and so on. So if there is a need, eventually, because we can't drop this and say, well, mRNA vaccines and this and that, we have to demonstrate using data. So if we show from data that mRNA is more superior than this, and then it has also a safety record, it's not rocket science to make mRNA vaccine. We can do that.
Yeah. Of course, clinical trials come with quite a number of challenges. So how what are, especially now, having experience working in Nigeria, what are some of the challenges you anticipate in the clinical trials, and how do you hope to mitigate those so that you can have them?
Well, I mentioned a couple of countries. The reason we're focused on Nigeria is because that's where we're based. So South Africa has well as established protocol and experience in conducting all phases of clinical trials, phase 1, phase 2, phase 3. We'll have just gone to South Africa, discussed, and said, do this. The strategy and incentive that we have is that any country that we do the clinical trials, they will be first in line to get the product. But there's no point going to do clinical trials and then you take the vaccine and then take it to another place. So that is attractive enough to get countries to be involved in clinical trials. So doing phase 1 trial in the US and in Nigeria is because that's where the factory, our own factory, is going to be domesticated. But that doesn't mean that we're not going to consider other countries. So phase 2 trials, for example, this is going to happen next month. Once the data is out, we will begin the clinical development, and that will require us getting all these parties involved, the Brazil, the South Africa, Nigeria, and so on. So right now, even the Saudi, Egypt relationship is all hinged on the data that we're expecting to come out next month.
What is the situation in regards to intellectual property and patent issues on the continent? Would you shed some light on that? Good
question. You see, what Africa should be pursuing now is not whether they should, companies or countries should relax patent or not. Okay, if the patent is relaxed for COVID, how are you going to produce it? Where's your factory? What are the people that can make this thing happen? So it's not enough if I give you the recipe. I presented, as you can see, I didn't hide the production process. I presented it to you. You can send it to anybody. Sending it to that person doesn't mean that you can produce it. I'm not, if I wanted to say I want to protect it, I'm not showing you the process. I've shown you how you're going to produce it. So patents and know-how are two different things. I mean, go down to your grandmother, for example. They make all kinds of nice soup, and then you want to make that soup. They give you the recipe, put this, put that, and you go and do, will you get the same value? They have to be involved in making sure that you get the right thing. So patent is one thing, the know-how is another thing. And in fact, even Moderna does not enforce their intellectual property this thing. So if Moderna, okay, the publication, everything is there. Can you make it? So it's not the issue of whether patent is patent that is the know-how. So the first thing that we need to do in Africa is to have that capacity first. I mean, look at the Serum Institute. AstraZeneca didn't hide there. This is the license the technology to Serum Institute manufacture this for us. So if you have a facility in Kenya, in Nigeria or South Africa, they can as well license that technology for you, I mean to you, to produce it for that region. You know, so that is not going to solve any problem. You know, and the danger is that it will rather kill innovation, because why will a company go and spend hundreds of millions to billions to develop a product where they cannot have control over? So this thing is tricky. We have to be very careful and then, you know, discuss this in detail.
It's interesting because, I mean, one of the many things that have always been proposed is the use of regional blocks to try and harness, you know, sort of harness the resources, because like you say, it takes a lot of time. It's a very costly affair to put up a vaccine manufacturing plant. And so I think, you know, like you rightly said, I think the approach of having, you know, economic blocks, you know, gather together so that then serves, you know, that particular region. And all that does, it has assured market. So, you know, that becomes a more interesting approach.
Yes.
Could the new facility also be utilized to produce monoclonal antibodies?
Yes.
Okay, interesting.
That's something that is very important, which you want to address. The regional blocks, the role would be to create the market. When you create the market, automatically the companies will come in to set up the factory because there's guaranteed market, but not for the regional blocks to say we want to have regional vaccine hubs. How is it going to work? The vaccine manufacturing is a business, is a private sector-led initiative. So governments will support those initiatives. I mean, in times of epidemic or pandemic, the government intervene by supporting the private sector, but not being in the business of the private sector. I mean, look at here in the US, Pfizer, Moderna, Novavax, all these people got funds from the US government. But US government is not part of the business. It's like, this is a problem for us. How can we produce this vaccine? Take this support and produce it. Government cannot be in the business of a vaccine. So that has to be clear. And until we get that, you know, clear, we'll go around the cycle. I hear we want to create this hub to do this, want to create. I mean, what is hub?
Yeah.
Who's going to man this hub? What are you going to produce there? What is the strategy?
Yeah. Absolutely.
So this thing is this thing of the private sector. The individual companies are the one that will eventually make this happen.
Interesting. I know this is a conversation that is very close to your heart, but unfortunately, because of time, I would like to bring it to a close. There are quite a number of messages of people just thanking you because of taking your time to discuss this with us. And also, as I join in and thank you very much for the presentation. And I know some of these conversations, the more, we constantly have to have them, through and through until we, we change the meager state of vaccine manufacturing on the continent. To everyone, thank you very much for attending. The presentation will be made available on the AVMI website, so you can always go back and clarify something that you would have wanted. I am sure also that if you do need to get in touch with Simon directly, Melissa will be very happy to facilitate introductions so that the conversation can keep on going, and then as we march towards doing the right thing in terms of our own being sustainable in terms of getting our own vaccine manufacturing up there on the continent. Thank you very much, Simon. Melissa, if there's nothing else, I'll try to bring this to a close.
Thank you, Charles. You may go ahead.
Thank you. Thank you very much, Simon, and let's keep talking.
Thank you, Charles, for the great moderation.
Thank you very much. Asante.
Okay. Later.
Thank you. Bye-bye, everybody.
Bye.
Thank you. Bye.